Hello, this is going to be a talk that is going to be looking at the basics of genetics that you might potentially need to know so that you can understand VDJ arrangement. So, if you have forgotten everything that you learned in genetics, never learned anything in genetics in the first place, or never took genetics, this might potentially help. Normally, when we're talking about the progression of any type of anything within biology, we're going to be talking about DNA to RNA to protein, right? So that is going to be the way, this is called the central dogma, and it's essentially the way that things happen. So we have transcription and then translation. Okay, this is how this happens, right? We have all of this happens in the nucleus. This happens in a ribosome, and that's how we do things. If it's membrane bound, then it's going to be in the rough ER. If it's secreted or otherwise, it's going to be out in the cytosol or the cytoplasm, depending on who taught you how to say the words. So what we're going to be talking about with this is... sorry. Is how this actually works, but then is also vaguely complicated by some of the things that we need to do in the immunoglobulins and T cell receptors, so BCR and TCR. We know that within our BCR and TCR, we have this highly variable portion that is going to be changing dependent on the genes that are going to be present, and that's essentially how we're going to get variability within these things. So we have this stuff up here, which I now realize is pretty much off the screen, but in any case, most of this is going to be conserved. So this is conserved. And we have our variability and our high variability, right? So how do we get that if we have DNA? And DNA makes RNA, RNA makes protein. How do we get variability? Okay, so we'll talk about that in class. There's other videos that are going to go over that, but in order to get to that in the first place, we need to talk a little bit about gene splicing. So we have our DNA. Our DNA is going to look something like this. And there's going to be part of our DNA that we're actually interested in, say from here to here. And your body knows more or less what to transcribe and how to transcribe it based on a large number of other different types of factors. Where the Tata box is, where all of these other different types of enhancer and promoter regions are, all of these things that are going to say, yes, this is the gene that you need to make and when you need to make it. We're not going to get into that. Within this DNA, there are going to be chunks of things that are introns and chunks of things that are exons. The introns are going to be the junk DNA in between, the untranslated parts. So the parts of the DNA that we really don't know about or don't really care about for that matter. And then we're going to have portions of it that are exons. And those exons are going to be the bits that get translated. At the end of the exons, there's going to be a stop. And at the beginning, there's going to be a start. And all of these other fun things that are signals within the DNA that will tell us when to make a protein and when not to make a protein. So, if we were to take all of this, when we're looking at our RNA is going to transcribe all of that. Our RNA polymerase is going to come along, and then we're going to get our RNA chunk that gets transcribed into exon 1, intron, exon 2, intron, exon 3. And within those exons, we still, so this is our DNA, this is RNA. Within those exons, so that's how transcription works, right? Within those exons, we still have this chunk, so our AUG will be here, and we have our stop over here. And that's essentially how we end up getting along with this. Now, to be able to get to all of that, there's going to be all of these different other types of things that are going to go on. There's going to be terminator sequences. There's going to be the Tata box that tells us when we actually need to transcribe. All of that fun stuff. Now, how do we get from this to the next step? Well, within these exons, there are going to be signals that are called splice donors and splice acceptors, because that's essentially how you say this is where the exon ends and this is where the next exon begins. If you have a splice donor, then that's your signal to say, we're going to go on. You need to stop, or you need to cut out the intron that's in between here. So we will have some sort of signal that will say splice. And then some sort of signal that says you're done splicing. And we'll have other signals that will do the exact same thing. So we'll eventually end up splicing out the introns so that we have this bit with an untranslated region that's technically called the 5' UTR, or 5' untranslated region. You then have exon number 1, and then exon number 2, and then exon number 3. Okay? So this would be our mRNA. We also add on a 5' cap and a poly A tail. Now the 5' cap and the poly A tail make it so that enzymes inside of your body don't eat it up. Because we know that just having enzymes or RNA around isn't a good thing and is generally going to lead to bad things happening, you know, like bacteria or viruses killing you and stuff. So we know that we need to be able to get to this point. So we have these different bits that get spliced out, and then from here we have our AUG and our stop, and then we'll make our protein. Look at that, it's a protein. And this is the translation that happens in a ribosome. And we end up making the protein that we need to make. This is simple genetics that you need to know about when we're going to be talking about how VDJ works. Because with VDJ, essentially what we're going to be doing is changing these different types of exons so that rather than going E1, E2, E3, we might go E2, E1, E3, E8, for example. And we'll get to that in class. Hopefully this was helpful. If it isn't helpful, or if you need any other assistance, please reach out, and otherwise I will see you in class. Bye.